So I'm very appreciative for the invitation, and I am anxious to present this information to this group. I haven't presented it before. This is new information and hasn't been published yet. We're getting ready to put that together. I'm always humbled when I follow a lot of really strong basic and translational research. As a clinician, the idea is to translate that into something meaningful to our patients. Every now and then, as a clinical researcher, I've tried to humble the basic and translational scientists to say that all of these algorithms and predictions and models haven't quite worked, and we have to try and figure out why. So we made a decision to take a very unbiased approach and think about one aspect of personalized medicine and developed a feasibility clinical trial, trying to think about how to tackle this problem in glioblastoma, one of the diseases that we treat. So as you know, the standard of care is really not a very good standard of care because it doesn't result in the cure of many patients. And in fact, once the patient has gone through their initial, quote, standard of care, there's no effective therapeutics, and almost all patients die within about a 9-month period of time from that point. And that's a real big challenge, and there are many reasons for that problem, and I think many of you know that, one of which, of course, is heterogeneity. And heterogeneity is a word that I learned about a long time ago, but I still don't really understand at all. And I think of heterogeneity, just as many of the scientists have talked about already, both individually and within groups, but also spatially within the tumor region of an individual patient, where we see dramatic differences just 1 or 2 centimeters away when you take samples from a patient. And then to have a personalized approach with actionable therapeutics based on a sample of tissue that actually represents a multitude of signatures, if you want to put it that way, develops a problem. But most of the time we reference this particular slide, which is where the largest clustering of potential actionable alterations are found, at least based on the TCGA. It's an old slide, but it has spawned a great deal of clinical research looking at a lot of different medications and drugs that have been tested, almost always in single drug therapeutics, which have not necessarily resulted in any clinical meaningful benefit. And the reasons for that, again, are legion, but single agents in a complex biology is unlikely to be very successful. And as Dr. Hood mentioned, to use one drug, I think, is unlikely to be helpful. So we have a plethora of drugs, some of which are, quote, positioned. They were designed to be used in this disease or for these particular targets. So we also have been fortunate to receive funding from Catherine Ivey and the Ivey Foundation, and about 4 years ago we started looking at different aspects of clinical trials research that may help inform the next generation of clinical trials. And after a series of studies that mostly were related to giving drug before surgery, looking at PK and PD relationships of those particular drugs, and like many years of research, always finding that there was drug concentrations in the tumor that was removed, but still no meaningful benefit, we decided to try a different approach. So the Ivey Foundation supported us, and we developed what's called the Early Phase Clinical Trials Consortium, which is a group of investigators around the country, largely senior investigators, and we came together and wanted to think about different kinds of paradigms and approaches. So at the 4-year mark of this consortium, the backstory is we gathered together with Catherine Ivey and researchers at TGen, that have done the profiling for us, and we started to think about what can we do to actually gain more information for discovery, again, that may inform next generation studies. And over the course of 6 months, with a lot of people around the room and on email and telephone conversations, we really had a complex conversation about how we would start to approach this, and it was difficult because there was a lot of bias about what one might do to achieve some sort of precision in therapeutics. And there was no consensus, I have to say. It's like the silos that you build internally, but externally there are a lot of feelings and strong opinions. But ultimately, we decided we had to start somewhere, and we decided on a feasibility study. So the story here was that we wanted to start the process of learning how to do personalized medicine in patients with glioblastoma. We chose probably the worst patient population, which are those with recurrent disease, and tried to take a very unbiased approach. So what we did was to make the decision that patients with recurrent disease who were eligible for surgery at any time in their disease, could be first relapse, second, third relapse, with any prior therapy, we would start a feasibility study to see if we can acquire good usable tissue, good usable RNA and DNA, and profile that in a way that was meaningful, that would define some potential for actionable targets, develop a molecular tumor board-based therapeutic strategy, and see if patients and physicians would accept that sort of recommendation. So that's the broad stroke of what the approach was, a very simplistic idea, and we would make therapeutic decisions based on any drug in the FDA pharmacopoeia, so positioned and repositioned drugs. As you've heard before, that there are a lot of drugs that are fairly cheap, that are used for many indications, that actually have a literature in glioma suggesting that they may be useful for targets like minocycline, mebendazole, metformin, disulfiram, all of those kinds of things, plus the other drugs that have been known to be targetable for glioblastoma, and then also made the decision that we wouldn't use one drug, we wouldn't use two drugs, we would allow the option of up to 4 drugs, so that we would take a complex biology and try and put together a complicated therapeutic and see if we could target all of the actionable aspects of the patient. In the same time, we had secondary aims, which were to develop xenografts in each of these patients, also to get liquid biopsies. Basically, we'd get blood for patients, looking at circulating tumor DNA as a potential biomarker, and then add to the database. So that was the idea. And there are a lot of challenges, and I mentioned them, one of which, of course, is the tumor heterogeneity. So we also thought we'd get a handle on that a little bit by taking the enhancing disease, and when Mitch would bring the patient to surgery, he would outline the region of the tumor that was enhancing, that was taken and used for the profile. But we also took disease that was outside the enhancing region as well, and in a secondary aim, used that tissue for the same kind of profile to see if there would be any differences, and we assume there will be, any differences in the recommendations or the actionable polymutations that would be seen within that. We also know that drugs sometimes work great in the dish and sometimes great in the flank, but not well in patients because of the pharmacokinetic restrictions of getting drugs across the blood-brain barrier. And we had to make decisions about drugs based on what we know about the physiochemical characteristics of those drugs and the pharmacologic characteristics of those drugs, and pick therapeutics that we hope would get across the blood-brain barrier, and again, using a multitude of drugs so that we wouldn't just use one drug, which is always proven to be unsuccessful. So that this doesn't use drug discovery, doesn't use synergistic complex therapeutic strategies. It's just basically seeing what we find, seeing if there's an action, and starting the process. So we put that together, and that was our strategy. And it was, again, only a feasibility. We weren't sure that we could actually do it, so we limited the patients that we were going to treat to just a small cohort to get all of the steps in place so that we could determine if we really have the potential for doing this kind of stuff. We also recruited a neuropharmacologist who scoured the literature about many of the drugs that are in the pharmacopoeia, looking for those that were, again, more likely to cross blood-brain barrier, and also had enough literature associated with them that we could refine that list as well. So this is the drug database that we came up with, and it's a unique database that was curated by the group. John Kuhn was actually, he's a neuropharmacologist in San Antonio, but I've worked with him for many years now, and also the group at TGen, and they curated the literature, they refined it, they created this drug database based on the general drug information that you see here, the clinical experience that was already known within a glioblastoma subtype, the effect on potential for blood-brain barrier penetration, whether it was an efflux pump or not, and would be associated with pharmacokinetic interactions and drug-drug interactions. So that was all put together into a database and was discussed at length, and we ended up with an enriched patient drug list of about 181 drugs that fit the criteria that we thought would be best. And of course, that, again, is our best estimate of what these drugs might be. And we allowed, again, positioned and repositioned drugs. We didn't want to be restricted just to those drugs that were produced, quote, for glioblastoma. So that was it. So the strategy is basically summarized here, where we would get an RNA and DNA profile. We'd used whole exome profiling and RNA-seq, both. We had normal for genomic DNA. We had blood. We looked at drugs first that may have the possibility of crossing blood-brain barrier. Then we developed the list of alterations, linked it to a literature, and then convened a tumor board. So the tumor board was basically compiled of all of the investigators at the IV sites. We would meet generally for 1 hour per patient. We would discuss all of the alterations, and we would go through the information that was presented to us based on the specifics of the enhancing tumor DNA and RNA for that specific patient. And we would go through the issues related, again, to the alterations, the literature, the blood-brain barrier characteristics. We would have an evidence report that would overview all of the alterations, and it would basically, we would try and rank them as best we could based on that literature and the evidence that was there. We had hyperlinks to the original data and the literature, so we could go quickly, just click on a hyperlink and go to the manuscript, the paper that discussed the specific preclinical or clinical reference to it. The blood-brain barrier report used a couple of different algorithms to support it if you're interested. One is the literature evidence, where you know patients have had drug and have had tissue taken, so there's at least some information that way. Something called a CNS MPO score, a quick pro CNS activity prediction, and an efflux transporter, an influx transporter interaction, and CYP450 interaction. So all of those were put together into a profile of those things. So the strategy was to do this in 15 patients with recurrent glioblastoma, to do the genome-wide whole exome RNA and DNA, to do the drug matching algorithms, and then make a report. the report actually when we talked about these things, first the TGen group would present the information about all of the alterations, including the allele frequency, the coverage, the details of that. Then we would talk about the CNS pharmacokinetics of the drugs that were matched as a group. We would talk about the patient in particular and their prior therapies, issues that may correlate with whether you'd want to use a drug or not, whether they were an alcoholic or not, for instance, and not to use disulfiram, or we could, for instance. Issues about prior therapy, their age, their performance status, concurrent medications, and prior history. So we would try and come up with the safest, most reasonable list of up to 4 drugs that might be reasonable for that patient. So in order to do feasibility, the first issue that we asked was, could we do it within a reasonable period of time? Because we can't wait for 6 months or 4 months to get the information. We set the parameter at 35 days, but we wanted to get all of this done with a written report that would be available, would be put into the medical record, because this is all CLIA. This is all CLIA-based. That would be put into the medical record and available for the physician to use to discuss with the patients. And also importantly, to have it as part of the medical record so that when drug companies or insurance companies get a request to treat a glioblastoma patient with metformin, propranolol, minocycline, and mebendazole, they're saying, what are you talking about? So we had at least some expert opinion, as best we could do that, to try and get drug companies to approve these drugs, in addition to an EGFR inhibitor, et cetera. So we were able to do this within, on average, 27 days, from the day of surgery to the tumor board recommendation that was actually printed out and signed by everybody. There was a range. We had to have usable RNA and DNA in order to have efficacy and to have that report. So we ended up enrolling 20 patients to get 15, and we actually, because of circumstances, had 16 that were eligible. The ineligible patients were 4 patients who had insufficient tumor content. There was mostly necrosis or reactive gliosis, or really wasn't tumor. And sometimes you can't tell, right? Because an MRI scan is not the greatest biomarker all the time. And then there was insufficient RNA from a couple of patients. But this is just the plot looking at the time. The patients that we treated were generally younger, because they were all treated at UCSF, and we tend to attract a younger patient, healthier patient population. But you can see more than half of them had been treated with multiple therapeutics over time, so they were in second and third relapse. There was a high percentage of methylated, MGMT methylated patients. I think that was just the skew of the 15 or 16 that were totally eligible. And they had all been treated basically with standard of care. A lot of them had been treated with bevacizumab and other therapies. In terms of the patient acceptance and the physician acceptance, because we didn't mandate that they get treated, we just offered the information. Patient acceptance was actually quite good. A large portion of patients accepted it, 47%. We didn't think that many would. Some of the reasons that patients didn't get treated were because of insurance issues. We just couldn't get the drugs that we suggested. But on average, those patients who were treated with over 3 drugs. The average was 3.4. So we actually were able to treat patients with a large number of drugs. And we learned a lot. So this is a learning process for us. And these are just 2 simple reports that we get. And I'll talk about these 2 particular alterations. But we were seeing a lot of different spectrum of diseases in terms of mutations and alterations. The average number of single nucleotide variants for all patients was about 56. So that's a complicated biology. And then we saw some of these hypermutated cases. We actually had 2, where we're talking about thousands and thousands of alterations that are present. So this is the data, and on the left side just shows the patients, and on the right shows the different kinds of alterations that we were seeing, and they're coded, based on actual mutations, high-level gain, low-level gain, fusions, etc. And you can see the spectrum of just in these 16 patients of alterations that were, quote, at a high enough level and felt to be actionable, that the number of combinations of alterations and approaches therapeutically could be quite large when you look at a list like that. But you did see, and I'll show you a little bit later, that they do tend to cluster into some groups. And these are the treatment recommendations that came from them when you look at it. You can see we used minocycline. We actually used palbociclib because of the CDK4 pathway, the RB pathway being altered. We used propranolol, metformin, minocycline, disulfiram, even CCNU. So there was a spectrum of therapeutics that were recommended based on the molecular tumor board recommendation and the alterations, blood-brain barrier characteristics, etc., that we found. And this is, to me, very interesting. It's in your face heterogeneity, but it's also kind of important that we get this kind of information. And this in relapse patients, right? It's a very heterogeneous patient population. And again, this is going back to the clusters that we saw about 60% of patients having alteration in the EGFR pathway. For instance, the RB, CDK4, and 2A pathway was altered in a large portion of patients. P10 was altered in that whole pathway in a large portion of patients. So you can see that we're starting to refine perhaps buckets that could be used for efficacy studies going forward, or adaptive design studies going forward, but they're starting to cluster. And again, this is just the enhancing region. We haven't even looked at the non-enhancing region. That analysis is going on right now. That non-enhancing area of tumor was about 1 centimeter on average to 2 centimeters away from the enhancing. So this is the enhancing actionable profile that we were seeing. We saw some unusual things. This was a fusion from the EGFR CEP14 that was described in only about 4% of patients in the literature. It was an interesting observation in the literature. Actually, there is data preclinically that if you use lapatinib in these patient-derived xenografts, that they would potentially have a good response to it. So this was a unique profile, and because the patient also had alterations in P10 and CDKN2A, for instance, we used an EGFR inhibitor. We recommended pulsed erlotinib, high-dose pulsed erlotinib for this patient, palbociclib for the CDK4. The patient was RB proficient, and mTOR inhibitor because of P10. This was another case of a hypermutated case with a lot of alterations in mismatch repair, MSH6, for instance, which has been described and has been suggested would be more susceptible to checkpoint inhibitors, including PD-1, and this patient was treated because of that with a PD-1 inhibitor. We have a couple of patients who are success stories of the 16-2 or long-term therapeutic, quote, benefit. They had some benefit to them, one with being treated with olaparib and trametinib, and one being treated with CCNU and metformin. Again, it wasn't an efficacy study, it was a feasibility study. We wanted to see if we could pull this off and do that. Probably the more exciting thing to us, besides the fact that we've been able to get this down into a short time frame to get good quality CLIA, RNA, and DNA-based information, is the secondary aims where, again, we're looking at the non-enhancing tissue and doing the same analysis. We're getting circulating tumor DNA, and we'll match the alterations from the tumor in the blood and see if we could detect at any threshold the same alterations. We get blood every 4 weeks in these patients to see if there are shifts that are occurring temporally over time. And then we're getting patient-derived xenografts, and this has been quite successful. We have the circulating tumor DNA on all of the patients over time, multiple times. We have all of the non-enhancing tissue, and that analysis is almost done. And then we have xenografts that are percolating in a good number of them. And so the goal of the xenografts is to do the iterative go back and look at the profiles to make sure they have the same fidelity as the patient, but also to test then the drug strategies that we assumed were appropriate and see if they actually work in vitro and in vivo, and then look at mechanisms of resistance. So the idea there would be that if they have a response in vivo, then over time they'll develop resistance, and then we can start to map out, some of the reasons for the lack of success in going forward. So that was it. It's just an early approach. I think we're trying now to develop an efficacy study based on the information of this. It's been very difficult to make that happen because you have to have a lot of patients. You have to have a control arm. Patients aren't anxious to be part of a control arm when they have disease like this. They don't want to be randomized. But those are the kinds of things that you have to do to get validation that the strategy might work. And I'll end there. Thank you.